Thank you very much. Nicole sends her regrets. She's applying to grad school and has an important meeting with one of her schools right at this exact time. So it'll be me giving the talk. But I've got both of our emails down there. So if you have any questions now, or if you're listening to this as a recording, feel free to contact us. So January is actually Cervical Cancer Awareness Month. So if you get nothing out of this talk, but these important facts, that would be great. Every year, over 500,000 women are diagnosed with cervical cancer around the world, and almost 300,000 of those women will die. The vast majority of those are in the poorest countries of the world. In the US, 4,000 women die every year of cervical cancer. This is actually tragic because this is a preventable disease. But as you can imagine, those 4,000 women tend to be in the poorest areas of the country, or women living in poverty without health care. The cause of essentially all of cervical cancer is human papillomavirus infections. And this is a virus that is transmitted by any form of sexual contact. And so virtually all of us will be exposed to this virus at some point in our life, but there's essentially no symptoms. You don't get a fever, or there's no outward way to know if an individual is infected. We do, for the last 15 years, have had a highly effective Hpv vaccine, and this is recommended for both boys and girls starting at age 11. It's approved up to 26, but actually older people can take it. And then we have mechanisms of cervical cancer screening, Hpv testing, pap smears, and you should consult your physician. The guidelines are changing now that individuals are vaccinated. Some individuals are vaccinated, so you should talk to your doctor about the best screening protocol. So here is the timeline of what happens over an individual's lifespan. Like I said, virtually all of us are exposed to this virus in adolescence or early adulthood, but the immune system clears infections. About 90% of infections get cleared within a year or two. So by the time you get to about age 30, for women, about 10% of women are going to be persistently infected. And it's those women who are susceptible to developing precancers, which essentially are also asymptomatic, and later cancers starting in age 30 and up. So luckily you can see there's a big time lag between appearance of precancer and cancer. And this is why the pap smear, which is really not a great test, it's only about 50% sensitive, works, because women typically would get a pap smear every couple of years throughout this whole span. And most of the time you would identify a precancer and treat it before it turns into a cancer. Obviously, way better would be to vaccinate with HPV before the age of when young people start getting infected. And one interesting thing I've recently just learned about HPV, and we're all hearing a lot about herd immunity, because the virus is transmitted typically from one person to another in a linear fashion, by the time you vaccinate only 20 or 30% of the population, you start seeing significant reductions. And so herd immunity actually takes hold in HPV at a fairly early stage. Now, obviously, HPV testing is important. If you're, say, a woman of, say, 30 and you're HPV negative, your chance of getting cervical cancer over the next 5 years is close to 0. And so that provides what's called a very high negative predictive value. And so now HPV testing is being recommended as the first, after vaccination, is the first step of primary prevention. Now, HPV does not only cause cervical cancer, it causes about half of all head and neck cancers and half of all anal cancers. And these are two of the fastest increasing cancers in high-income countries. And it's predicted that shortly, more men are going to die, say in the US or Europe, of HPV-driven cancers than women, largely because, at least head and neck cancer, there's no good screening test for. And so here you see on the top the incidence of head and neck cancer, which is slowly rising, unlike most cancers, which are declining, and mortality has pretty much stayed flat. Anal cancers as well are increasing in prevalence and also increasing in mortality. And for reasons we don't understand, men are more susceptible to high-risk HPV infection in the oral cavity. And so they get more HPV-driven head and neck cancers than do women. anal cancers are pretty much a little bit more prevalent in women than men. Now, across the world, as we mentioned, this is over 500,000 cases a year and over 250,000 deaths per year. You can see in the Americas, regions in Central South America with very high incidence. The US appears like a uniform green of low incidence, but we'll see in a minute that that's somewhat of an illusion. And then there are over 100 types of human papillomaviruses that can infect our body. There are 15 types, 15 strains, or in the HPV literature, we call this types, that can cause cancer. By far the most prevalent, as you can see, is Hpv 16, which is the most oncogenic form of HPV, and it causes over half of all cancer. And this holds pretty much around the world. Everywhere around the world you look, Hpv 16 is the most prevalent type. And then the prevalence of these other types varies depending on where you are in the world. So here is the incidence of cervical cancer across the US, and you can see that there are areas of the South and through Appalachia all the way to New Jersey, which have higher incidence. If you break this down by medically served region, you can also see that there are hotspots where there are considerably more cancer. Undoubtedly, these are areas with higher levels of poverty, lower levels of access to medical care. And to put this in perspective, in 2020, it's estimated that as many women died in Asia and Africa of cervical cancer caused by HPV as died of COVID-19. And so even though we've had an HPV vaccine for 15 years, and we only really need to vaccinate a third of the adolescent population, we have not achieved this in most of the world. Partly driven by this, I started about 10 years ago a cervical cancer study in the largest cancer hospital in Guatemala. And these two amazing women and physicians in the hospital recruited over 700 women into this study, collecting tumors, blood samples, clinical information, and we'll hear some results from that study. So first of all, let's talk about why is HPV such a dangerous virus. It is because it encodes in its genome two very powerful oncogenes, the E6 protein, which inhibits TP53, and E7, which inhibits RB1. So this virus can impact essentially the two most important cancer pathways in the body. HPV infects and replicates in the nucleus as a double-stranded DNA circle of about 8 kilobases. Integrating into the human DNA is not part of its normal life cycle, like for instance, retroviruses. This is actually an aberration that happens during the process of HPV leading to cancer. Many of these integrations are of a simple form where a fragment of the HPV genome containing the two oncogenes integrates somewhere into the human genome. This integration usually deletes two regulatory proteins, E1 and E2, which are involved in replication of the virus, but they're also involved in regulating the transcription starting from here, which transcribes the two oncogenes. So at least the dogma is that HPV integrates, deleting the E1 and E2 genes. That leads to higher expression of the oncogene, and so integration is favored and happens in most tumors. We're going to learn, though, that there are some major exceptions to this rule. So one of those is that HPV can also lead to a localized amplification where there are multiple copies of HPV in the genome, and often multiple copies of the DNA, the human DNA that was flanking that in a very complex rearrangement. And so there are actually three competing models about how this happens. One is a re-replication model where there's a simple HPV integration. The origin of replication is actually near where the promoter is, so it's present in essentially every genome. If a cell continues to express E1 and E2, which are involved in replication, you could have DNA synthesis happening in this origin of replication, while the virus is in the human genome. And so this would obviously be a very bad thing for that chromosome, would create a mess and could potentially lead to this kind of structure where you amplify the virus and local human DNA at that region. But there's another model, David Seimer from whole genome sequencing of a number of tumors came up with this model where you have double-stranded breaks, HPV joins at the end of that double-strand break, and then loops around and then replicates itself in the flanking human DNA, which then lays down in the genome. And like I said, these are sort of competing models. In vitro, we know that this happens. If you have an integrated HPV and you express E1 and 2, you can get amplification. And then there's a third model, which I'm not showing here, where instead of these circles laying back down in the genome, they can actually pop out and be replicated as extra chromosomal elements. So anyway, to kind of resolve all of this, this is why we got into using long-read sequencing and got involved in sequencing with the Oxford Nanocore platform. So first of all, what is a nanopore? As the name implies, it's a very, very tiny pore. It's embedded in a electrically resistant membrane, and electrical charge is put across here so that current ions can flow through the pore. It's just big enough for a strand of nucleic acid to flow through the pore. And so when that happens, the current going through the pore is disrupted. And you get this trace called a squiggle, which is actually dependent on the DNA sequence that's flowing through the pore. So native DNA, which is not modified in any way, as it's passing through the pore, one can deconvolute this electric signal and generate a DNA sequence. Now, the really key technological breakthrough was actually the development of a motor protein that would guide the DNA through the pore at about 400 base pairs a second. And this is slow enough that a computer can collect all this data and read the sequence. So this is really quite amazing technology. In its smallest iteration, this is a flow cell sitting in a sequencer hooked up to a box, which is a computer, which you could hook up to a laptop, or they've got a device where this flow cell sits into something about the size of a laptop. And so one of the attractive things about this is the whole thing is portable. You can take this sequencer essentially anywhere in the world. The data also comes out in real time. And so the computer is reading the sequence as it's going through the pore. And so there are mechanisms where you could actually be looking at the sequence as the sequencer is running and get data literally within minutes of loading the flow cell. We prepared and loaded various sizes of DNA, sheared DNA, which typically gives you the most output per flow cell, unsheared DNA, which gives you longer fragments, or ultra high molecular weight DNA, which can give you very, very large fragments. The MinION flow cells, you can load in a library if you've either gotten enough data, or typically after one day, the pores sort of get clogged. You can flush that out and load either the same library if you want more data, or load a different library. And typically in this flow cell, there are about 1,000 to 1,500 pores, like I said, which are collecting data at 400 nucleotides per second. So some of the advantages, there's essentially no limit on the DNA length. And so the accuracy of the sequence basically does not depend on where in the fragments you're sequencing. And so the only limitation to the length of sequence is physically how big of a piece of DNA can you get into the flow cell and get through the pore. The world record is 4 million base pairs. In our lab, we've gotten sequences close to 2 megabases. Like I said, the data comes out in real time. In many of the cases, we're isolating DNA directly from cells or tissue and sequencing directly. So modified bases can actually be called because they'll give a different signal as they go through the pore. So methylcytosine gives a different signal than cytosine. So does hydroxymethylcytosine. RNA can actually go through the pore, and we're actually getting ready to directly sequence RNA in the next week or two. And there are modifications of RNA like 6-methyladenine, which I'm just learning about that we're interested to follow through. And for us during the pandemic, all of this being self-contained meant that we could do all of this in our lab. Grow cells, make DNA, make libraries, load the sequencer, collect the data, and we didn't depend on a machine in another part of the building or people to run it. And so that has been a big advantage to us. Some of the challenges is that the raw read accuracy is lower than, say, short read sequencing, but Oxford Nanopore has done an amazing job at improving the algorithms to call the sequence. And so data that we collected 2 years ago can be recalled by newer software and we get more accurate data. The cost, depending on what you're doing, can be more than a similar project on the PacBio, which is the other platform out there for long read sequence. So you should really look at both depending on what your application is. So we have tested a large number of different methods on the Nanopore. It's essentially all we've been doing for the last year and a half. Like I said, we've done different size preparations of DNA. There are two different methods for targeted sequencing. One is to cut with CRISPR and add sequencing adapters where CRISPR cuts. And so you can do this to sequence specific regions of the genome, or in our case, viral genes. There's a method called adaptive sampling, which I'll talk to you about later. The standard methods involve ligation of a sequencing adapter to the end of DNA, but there are also transposons to add adapters, and I'll tell you why we might want to do that. You can multiplex samples by adding different barcode adapters, either as ligation or during PCR. We've done a couple different versions of transcriptome sequence, either direct cDNA protocol or a PCR cDNA protocol. And then analysis, we've used some cloud-based system that Nanopore has called Epitome. And the reason I'm here, we've used the Cancer Genomics Cloud. And we've also used some pipelines that have been developed either by Oxford Nanopore directly or by users. There's a very good user community. People that develop software to analyze this data are putting this software out on GitHub that anyone can play around with. We've begun depositing some of our data into a repository, and we have a preprint showing some of this data. We are probably a good example of, I guess, what I would call a command line challenge laboratory. I have one person in my lab who does some command line computing, but I don't. And so mostly we're depending on either bioinformatics people who are hard to find and are busy, or cloud-based mechanisms. And so the Cancer Genomics Cloud has been nice for us as a place where we can upload data. There are pipelines and programs already there that we can run the analysis, collect the files. So what files come off the sequencer? There's a FAST5 file, which is a raw data file. This can be stored and recalled in a future time to either for new applications or calling modified bases. After aligning to, then there's a FASTQ file, which is a text file of the base called sequence. And then there are various information files giving you the size of the read and other data. One application we obviously do is aligning our sequences to the human genome. And like I said, we've used this Epitome tool from Nanopore, which is a cloud-based system where we can point to the folder where our files are. They will get uploaded, taken through a number of sort of canned apps that they have, and give an output. And that, we've had sort of mixed success with that. So we've also been uploading merge FASTQ files into the Cancer Genomics Cloud, aligning with some ONT pipelines that are on the cloud, using a program called Minimap2 to map to the genome, and then standard, SAM tools to generate BAM files that we can view the data. We also obviously want to align to the human genome, and so we've taken the same protocols, just given a different alignment file to FASTA file to align to. And we've then used that, we have aligned separately to the human and HPV genome, and then cross-matched those outputs to find reads that align to both human and HPV. And then, as I mentioned, on GitHub are a number of pipelines to run and look for modified bases, to analyze transcripts for transcriptome or rearranged transcripts. To test out all this technology, since this was new, I mean, we didn't know of anybody that was applying long-read sequencing to HPV integration. We first turned to one of the cell lines that's been around since the 1980s, which is very well characterized, called CASCI. This cell line is very complicated. It has 800 copies of HPV integrated in chunks of HPV in 30 or 40 sites in the genome. And so it has been known since literally the 1980s that there's many copies of a full-length version of HPV integrated into this cell line, as well as a copy that has a deletion of about 1.5 kb. And so it was known that there were multiple copies of both of these genomes in concatenators in the genome, but nobody really knew what the structure of this was. Plus, David Simer's lab did a 30x whole genome sequencing with short read sequence, and it mapped most of the junctions between HPV and human DNA. So we thought this was a great test case. And so we did nanopore sequencing. We identified reads that had human DNA and then HPV DNA, and could start looking at the structure. Where was HPV integrated? And we found virtually all of the integration sites that were previously published. But then we could begin looking at the structure of these concatenators of HPV that are attached to the human genome. And we found that they are extremely complex. So these blue A's are the full-length version of the genome, and these yellow B's or orange B's are this specific truncated version of the genome. And essentially all these A's and B's are the exact same sequence. Then there are fragments that we label here at X, which are various different sizes of scrambled pieces of HPV. And try as we might, trying to align these different blocks of HPV to each other on different chromosomes, we couldn't see any pattern. Like there's essentially, these things got scrambled and then inserted into the genome. So this was to us, very interesting. Nobody had seen this kind of sequence before, and we're curious as sort of how this happened. We reasoned because it's exactly, the A's and B's are exactly the same, but their arrangement is different on different chromosomes, that these blocks of DNA must have formed before integration. So as extra chromosomal elements, which then integrated into the genome. And because we're in the era of COVID, we called this phenomenon Hpv Superspreading. Now, this is obviously only one cell line. And so, is this the case? And so the idea would be then that you have a 8 kb episome, which is replicating. Somehow it's being converted into a much larger structure with rearrangements, replicating extra chromosomally, and those different fragments then integrate into different portions of the genome. And so that scrambling happens before integration. So that was our model. Obviously, we need to, we speculate that there's this intermediate form between the episome and the integration of all of these clusters of DNA. So we found another cell line that also has multiple copies of HPV integrated in multiple sites of the genome, and we found a very similar situation. There are some full-length copies. They're actually pretty rare, but then there are two other very specific deletions. A version of the genome that has 163 base pairs deleted, and another version that has that same deletion and a second deletion of 367. And so we only find the 367 base pair deletion on top of genomes that have the 163 base pair deletion. And again, we found clusters of these HPV genomes seemingly scrambled in different orders. Sometimes there's 4 copies of this thing next to each other in one, sometimes there's 2. Very similar to what is happening with CASCI cells. Plus, like I said, we only find this double deletion. We only find this 367 base pair deletion when the 163 is present. So we reason that there must be a progression here going from a full-length copy to the 163 base pair deletion, and then to the double deletion. And we reasoned again that this must have happened before integration. This must have happened extrachromosomally. I'll come back to this later. The 163 base pair deletion actually deletes a critical repressive transcription factor binding site in the promoter. So we think that it's no accident that that region of the genome was deleted. It was actually deleted to give you higher expression of E6 and E7. So now, having convinced ourselves that the Nanocore sequencing was working and we're getting accurate data, and remember, we're isolating DNA directly from the cell line and putting it straight on the sequencer. So when we see rearranged fragments of DNA, there's never been a PCR step. There is a ligating adapters onto the end, but we believe that these structures are in fact intact. And so we sought to answer an important question. Remember, I told you HPV is the most oncogenic form of HPV that there is, and it doesn't always have to integrate to cause cancer. So we wanted to look into this. By contrast, HPV 18 and 45, which are also highly oncogenic forms of HPV, integrate virtually 100% of the time. So it's like they have to integrate in order to cause cancer. But HPV 16 and a related virus, HPV 31, integrate only about 2/3 of the time. And in fact, in a group of Guatemalan tumors that we published on a couple of years ago, we found this is kind of equally split between 1/3 of tumors which have only integrated HPV, another 1/3 that have no integration, no evidence of integration at all, and another 1/3 that are sort of in between and have a mixture. So again, we wanted to know, how is it that HPV 16 can cause cancer without integrating? What is it doing? So we were fortunate to find a cell line that had been established in Korea about 15 years ago and apparently forgotten about, that the authors reported had only episomal HPV 16. So this is very interesting because there are essentially no other cell lines like that. There are a few other cell lines that have episomal HPV for a time, but as you grow them in culture, the virus integrates and the episome is gone. When we did whole genome sequencing, we found that in fact, there was a piece of HPV integrated into chromosome 11. It's only a tiny fragment, 150 base pairs, not big enough to encode any of the HPV proteins, but interestingly, it's associated with an amplification of a region of chromosome 11, which contains a very interesting gene called YAP1. Now, YAP1 has been in the TCGA cervical cancer study, is in fact amplified in about 15 to 20% of cervical tumors. So it's one of the most frequent copy number variations. In this group here in 2019 showed in mice, you can cause cervical cancer by overexpressing YAP1 in the absence of any HPV proteins. So YAP1, for reasons we don't understand, seems to be a very powerful cervical cancer oncogene. So we think that HPV integrated into this region, caused this amplification, and most of the HPV integrated sequences were gone, but it left behind this amplified oncogene and contributed to the formation of tumor in this tumor and also in this cell line. The rest of the HPV sequences in this cell line are mostly in the form of HPV-only fragments, that some of which are 50, 100, 200 kilobases in length. And so here's aligning all of this to the HPV genome. All this sequence here with this colored is soft clipping sequences that don't align to the reference and so are hanging out the end. You can see insertions in here that are almost exactly the size of the 7.9 HPV genome or multiples of 7.9 kb. And we also see this 632 base pair deletion that's present in about 10% of the fragments. So this is very complicated. This sequence was actually generated by the standard ligation, where adapters are ligated onto the end of linear DNA molecules. But we were worried, of course, if these things are episomes and are circles, obviously, if you have a very big circle and you extract DNA, it probably gets broken up. And so you can ligate adapters onto the end. But if they're still intact circles, they would not get sequenced by this method. So we turn to another method, which is it's a standard Nanocore protocol, where a transposase complex can go in, cut the DNA, and add sequencing adapters at the same time. So you can have either very, very long DNA, and the transposon will add adapters, somewhere in the middle of that fragment. Or if you have a circular DNA, the transposon can go in and add adapters onto a circle. And so now you can sequence circular DNA. Now, to be clear, in this protocol, you're going to sequence both circular and linear DNA, but at least we don't lose the circles. And this protocol has no ligase, so that the sequences we get, there's essentially no way, if they're rearranged, for that to be any kind of artifact, because at no point have we introduced a ligation step. So when we did that and aligned, the soft clipping is gone here, that's why you don't see it, but you see alignment across the HPV genome. We see the same 634 base pair deletion. And we also began to see molecules that were essentially exactly one monomer of HPV or a dimer or multimers. So consistent with the idea that HPV normally would replicate as a single monomer, but somehow, at least in this tumor cell, can start forming dimers, trimers, and long-range multimers. And then also multimers, which have no human DNA attached to them. So these, we believe, are completely extrachromosomal, which have the full-length copy of the genome, this 634 base pair deleted version of the genome, as well as other fragments. So here we're seeing the intermediate that we speculated before. And we can see that HPV can go from a single monomer to a multimer structure with rearrangements happening, all of this extrachromosomally in all kinds of scrambled forms that then, if we're to integrate into the genome, would generate this super spreading as we saw in those two cell lines. Okay, so next we wanted to ask the question, does all this happen in actual tumors? So far, we're only looking in cell lines, very interesting cell lines, but does this happen in vivo? And so we kept with the tagmentation approach because we wanted to make sure that we would be able to capture circles. We had, somewhat limited amount of DNA from our tumors. So we would pool, say, a group of 6 tumors together with barcoding onto a single flow cell, carry out sequencing aligned to human and HPV genomes. And we took from our previous study, either tumors we knew were only episomal or tumors that were episomal plus integrated. And there was about 30 of each that we sequenced. Now we saw something very interesting, actually similar to this new 1000, the episomal cell line. We saw HPV reads that were almost exactly one genome. Usually there's 5 or 10 base pairs missing. And so I feel that during the transposon going in there, there may be a few base pairs lost. And so here's a genome that starts at position 202 and goes almost all the way around, or we had genomes that started here or here or here and went all the way around, essentially looking like randomly across the genome, you had circles, the transposon is going in, adding a sequencing adapter, and we get a sequence of that nearly perfect circle. Now, of the tumors that we knew had only episomal HPV, about half of them were in this class where we could only see monomers. We only saw these individual circles. And so it looks like in those tumors, that's all there is a seemingly normal circular monomer of HPV. So in fact, that shows, that HPV can cause cancer, as at least HPV 16 can cause cancer being in the cell as a monomer without rearranging or without integrating. The other half of the episomal tumors, in fact, had multimers or rearranged versions of the genome. Now, we had known previously that these episomal-only tumors were almost all squamous cell carcinoma. Normally, about 10 to 20% of cervical cancer is adenocarcinoma, which is a more aggressive form of the cancer. So these episomal-only tumors are a less aggressive form, and were mostly occurring in women shown here in pink, that are diagnosed with their cancer after the age of postmenopause or after the age of 45. So a somewhat later diagnosed, less aggressive form of cancer is associated with these forms of HPV 16 that do not have integration. Now, when we looked in the rearranged tumors, in episomal-only tumors that had rearranged versions of HPV, so we would see, so here's a tumor with a block of DNA, HPV DNA, that ends at position 6924 and starts back up again at 2475. So there's a rearranged junction here that's HPV to HPV. These blue asterisks are rearrangements that are happening inside either the E1 or the E2 gene. If you remember, these are the two genes that are virtually always disrupted during integration. Here we don't have integration, but we have interruption of the same two genes, either by having deletions in them or rearrangements. So to ask the question, how does HPV 16 cause cancer without integration? One way is to rearrange the episome, deleting the E1 and 2 genes. So instead of integrating and deleting E1 and E2, you just do that in the extra chromosomal space. Now, we also saw tumors that had like a 16 kb read that was almost exactly 2 copies of HPV that went all the way around one version of the genome and then another version of the genome and would end back almost exactly the same place. Again, sometimes we'd be missing 10 or 20 base pairs at this end here, but it looked very much like there was a dimer that got sequenced. In one particular tumor, all of those dimers had a deletion, either a 58 base pair deletion in one of the 2 copies, or 2 copies of the 58 base pair deletion, or a 58 and a 30 base pair deletion. Where are those deletions? They're exactly on top of where we saw the other deletion in one of the cell lines, and neatly take out the YY1 transcription factor binding site, which is this repressive binding site. And so deleting that site would in principle lead to higher expression off of this promoter and higher expression of the oncogene. So this is very exciting. It says that a second way that HPV 16 can cause cancer without integrating is to delete key binding sites repressing E6 and E7. So now you can have intact E1 and E2, but you delete a site that's normally involved in repression and get a high expression of the oncogenes. Now we then turn to this second class of tumors, which we've known from previous result had both episomal and integrated HPV. This is a group of tumors that are much more aggressive. Half of these are adenocarcinomas, which I said is a more aggressive form of cervical cancer, and almost all of them, shown in the pink here, are diagnosed before age 45. So early onset, more aggressive disease associated with this phenotype of having both episomal and integrated forms of HPV. These sequences looked very complex, where again, we could have multiple copies of full-length genomes, so multimers, but a lot more scrambled pieces of DNA. And again, where we saw junctions between one piece of HPV and another, almost invariably, we were inside either the E1 or E2 gene. So you have multimers of HPV, you have disruption of the E1 and E2 gene, and a very complicated situation. So a third mechanism of generating cervical cancer without integration is to generate very complex rearrangements of HPV. And these, as I said, are associated with early onset of disease, a more aggressive form of the cancer. And so obviously there's something we want to study further, exactly what's going on here. Now I want to turn to a very, very interesting application of the Nanocore system. Each of the nanopores is in an area which is electronically controllable. And so each of these pores can be controlled by the computer running the sequencer individually. So 2 groups independently, one in Hopkins and one in England, got the, to my mind, crazy but exciting idea that as the DNA is going through the pore and the computer is reading the sequence, can we have the computer match that sequence up to a known database and select what it's going to sequence? And so if the sequence going through the pore matches a region of interest, you just let it continue. You do nothing. All that strand will go through the pore and you collect that piece of sequence. If that sequence going through the pore does not match a region of interest, you actually reverse the current across that specific pore. You spit out the DNA that's in there. It actually blows apart, so it can't be sequenced again, that specific fragment. And then the pore is empty, it can sequence another fragment. And so this is a way where you feed the sequencer the entire human genome, for example, but you feed it a FASTA file and a bed file and say, we want to sequence only certain regions of the genome, only certain genes. In our case, we wanted to sequence, obviously, HPV selectively, plus certain regions of human DNA. And believe it or not, this actually works. Meanwhile, we were also very interested to try out a protocol to generate ultra-high molecular weight DNA. Now, this uses magnetic discs made by a company called Circulomics that PacBio just bought up. And so you can gently lyse cells in the tube, put in this magnetic disc, all your DNA will bind to that disc. You collect that disc at the side of the tube. You can then wash that disc and the DNA as much as you want, get it clean, put it back in solution. So at no point do you pipette the DNA, at no point do you precipitate it. And unlike magnetic beads, where a very long piece of DNA binding to two beads would probably get sheared, here, everything is all hanging around this disc and gets collected together. And so this allows you to produce megabase length fragments of DNA. And so Nicole in my lab said, well, that's all great, but we want to selectively sequence HPV in these specific regions of the genome. Can we combine adaptive sampling with this ultra high molecular weight DNA? So I thought that was an interesting idea. I called up my friends at Oxford Nanocore and said, hey, does that work? Can you do that? And they said, good question. We've never tried it. Why don't you do it and let us know what you find? And so Nicole did that. We built libraries with ultra high molecular weight DNA with Caskey and SNU 1000 and ran the exact same DNA either with or without this adaptive sampling. And you can see we get a big increase in the number of either human HPV junction reads that we find or HPV only reads. And these reads are huge. The largest read we found in SNU 1000 was 800 kilobases, and we found a read in Caskey cells close to 2 megabases. And the longest HPV only reads we got were 250 kb. The longest human HPV reads we got were 350 kb. And so with this, one of the things we're able to do is assemble some of these human HPV junctions in Caskey cells and find that we can go from human to the HPV block back to human, or from a large HPV block to a small piece of human DNA that's embedded inside there. And so we can start figuring out what is the structure of some of these very, very long regions of DNA. And we also applied this to SNU 1000, which has the episomal DNA and could get much longer reads to give us the structure of those fragments. So in conclusion then, we're discovering that HPV has a lot of tricks up its sleeve. It can integrate as a single copy of HPV stuck into one region of the genome, turning on its oncogenes. It can undergo these complex sets of rearrangements, generating multiple copies of HPV, all integrated into the same region of the genome. What I didn't tell you was HPV has a very powerful enhancer in it. And when you have multiple copies of that enhancer in one region of the genome, and often these sites are in regions where there's a human super enhancer, now you get super enhancer activation of both the HPV genes, but potential also flanking cellular genes. And we do find at almost every integration site, there is a local human gene that's activated that we can make a case has a role in the transformation of the cell. But as I've shown you, HPV can also cause cancer, at least HPV 16 can. without integrating, with either expressing simply as a monomer, which might have a deletion in it, causing some kind of rearrangement, or what I haven't shown you from transcriptome analysis, we can show that the oncogenes can be epigenetically activated in those monomers. Or you can have these multimer versions of HPV, either the normal genome or deleted and rearranged versions. And then in some circumstances, those multiple rearranged copies can integrate back into the genome. So this is all very complicated. This is all very new. And essentially, we could not figure this out without this long read technology and without the Cancer Genomics Cloud to help us analyze the data. So then the last mechanism we have for HPV 16 causing cancer without integration is to keep the episome, so to keep the normal 8kb episome, but epigenetically turn on E6 and E7 oncogene expression. Exactly how did this happen? We're not sure. But one of the things we can do, as I mentioned, you can read modified bases off of Nanocore sequence. And so this shows 3 different sites of the HPV genome, which are CPG sites, which have methylcytosine modifications. And we did bisulfite conversion and Sanger sequencing of the exact same DNA, and we get essentially the same result. The beauty of the Nanocore result is that we have this DNA, we have this data across individual sequence reads across the entire genome. So we can look in very great detail as what's happening to, for instance, methylation of HPV DNA, either before integration or after integration, and that's going to lead to a lot of exciting results. Now, I'll just end with the idea that mobile sequence elements obviously are not a new concept in biology. Barbara McClintock discovered these in the 1930s and 40s, studying maize. We know this also happens in cancer. So when you select cancer cells for drug resistance or resistant to a particular therapeutic, you can get the formation of amplified extrachromosomal DNA elements called double minute chromosomes. And these can exist either as extrachromosomal elements, or a number of these can all join together and be integrated into a specific region of the genome, something called a homogeneous staining region. So we know this happens in cancer, that oncogenes or drug resistance genes can get amplified and then inserted into the genome. And so a very powerful parallel thing we think is happening in HPV, where you have this extrachromosomal DNA, it can amplify, and then it can also integrate into the genome. And our hope is that understanding how this process all works in HPV will also help us understand how cancer cells are regulating genome. One thing I didn't go into, there's a whole new field of extrachromosomal DNA in cancer. And so maybe 20 to 30% of all tumors have some form of extrachromosomal DNA, often having an oncogene in there, and it's very open chromatin, very high expressed. And so there's a lot of interest in studying how does DNA pop out of the genome and replicate extrachromosomally. And some of these long-range sequencing methods are going to be instrumental in figuring that out. So during the pandemic, I've had to train students. We had summer students who were all remote. And so one of the things we have been looking back at some of the historical giants in the field. And so we've gone back and studied some of the work of Barbara McClintock. And it's really amazing to read what she was able to do. Before the sequence of DNA was ever developed, she figured out that chromosomes were rearranging, that extrachromosomal elements were being formed, and discovered mobile DNA without ever having elements without ever having sequenced DNA. There are a number of challenges on the data level, so people are familiar with CGC. We still need some help, I think, in figuring how to upload our data. We're currently using, we know enough Unix commands now to actually use the cat command to join all our FASTQ files together and upload that file by dragging and dropping it. But we would help to be educated in other methods. It would be nice if we could have a text file output from the alignment. So if we could have a single text file that showed us for every read, did it align to either human or HPV? and then we could begin cross-matching those data sets. That would help us. It would be great if more of the nanopore-specific programs were on the CGC to use. Specifically, there's one program called Megalodon, which is calling on modified bases. That would be very useful to have. I'm sure there are transcriptome tools we could use with our existing data to help us to map transcripts to genes, look at alternative splicing, hybrid fusion transcripts. We're also generating data. Hi-C is a chromatin structure-based method, and there's a derivative of that method called Pore-C adapted to the nanopore. And because there are longer reads, you can actually see higher order structures. And so we're generating that data, but we're so far lost in sort of how to analyze that data and display it. And so it would be great if there was more tools using that would be more user-friendly. This is the site where Megalodon is, which is publicly available program. I'm sure it would not be a problem to put this onto CGC if that were possible. And this is the team that's done all this work during the pandemic, wearing masks, staying socially distanced, getting vaccinated and boosted. And like I said, one of the great things about this system is that we've been able to take each individual, do individual steps and carry this out in the lab. And I actually went back into the lab and did all the poly-A RNA isolation, have done some loading of sequencer, as well as the processing analysis. And then here's other individuals that have contributed to what to the data I showed you. So I will stop there and I'll be happy to answer any questions.
Thank you so much for an excellent talk. I'm so impressed you managed to cover technology and biology and clinical stuff and cell cultures and so many different things so coherently in one talk. And clearly the impact of the work.
Computing.
So if you have questions, please put them in the chat and we can ask. We've got a few questions already. The first one is, have you looked at the distribution of HPV integration sites across the genome? And is it random or localized to specific chromosomal regions? And do you know which ones, if it is localized?
Yeah, that's a very good question. Initially, when people first started looking at HPV, it seemed like it was totally random. Then people started seeing some integrations around some oncogenes. So for example, HeLa cells, HPV is integrated near the MYC oncogene. And in fact, we know that it amplifies MYC. And that's probably part of the reason why HeLa is one of the most aggressive cell lines on the planet. And as we've gotten more and more data, we have seen specific integration sites. Actually, Alison McBride has an excellent preprint on bioarchives where she's looked at this in detail. And what she's seeing is that HPV often integrates near where there are super enhancers. And so we think that that is being selected for, that HPV will integrate near a super enhancer, both to enhance the expression of its oncogene and potentially also to turn on those super enhancer sites, which are known to be involved in stem cell activation in cancer formation. So we think the actual integration is random, but then what is selected for are HPVs happening in certain sites, which either allow the oncogenes to be highly expressed or certain local human genes either to be knocked out by insertion or to be turned on by integration.
Very interesting.
So some of those are the MYC oncogene, the RAD51B, TP63. So there are a bunch of known cancer-related genes that are often integrated nearby and activated or inactivated.
Cool. So on the CGC, which tools were on the CGC already and which did you have to put on? And kind of what was the process like for doing that? And then I guess there, what tool are you planning to add tools in addition to that, like the Nanocore ones you mentioned, or are you encouraging the Nanocore community to start working on getting more cloud available?
Some of both. We're probably not talented enough to add tools ourselves, but we would be delighted to work with others that could help mount tools and then we could test. So we got started because one of the Epitome app generates a whole bunch of BAM files. So we have hundreds of folders of FASTQ files, and it will generate a BAM file for each one. So we were uploading those to CGC and using SAMTools tool to merge BAM files into one, and then index to get a index file. Then we could look at in IGV. There was the Broad actually uploaded an ONT PacBio pipeline, and so that's actually already on the cloud. And so with Dave Robeson, he taught us how to use that to bring in our version of the genome or to bring in the HPV sequences we wanted aligned to that, and then that generates a merge BAM file that we can then look at. We're just starting to play. One of the interesting things that's on CGC is if you generate a BAM file, I know you have some tools to directly look at the BAM file on CGC, or you can get a link that you can open in IGV, and so you don't have to download that massive file onto your computer and have it slowly loading. You can actually, through the cloud, look at your BAM file stored on the CGC. So I think that's an interesting app. And actually, I would like to learn more about the tools that are already there to visualize BAM files within CGC. So if you could point us to a tutorial on that, that might be interesting.
Yeah, we can definitely follow up on that.
So then, like I said, I mean, we're generating a lot of transcriptome data. I would imagine it's very straightforward. I mean, that's just RNA sequence transcriptome. I imagine there are a number of tools already on CGC to align transcriptome reads to genes or to look for transcripts, alternative splicing. So that shouldn't require a Nanopore specific tool. And so that might be the next thing that would be the easiest thing to do.
Yeah, that makes sense. We have a question about whether you've had a chance to compare HPV positive who do not have cancer samples with cancer samples who do not have the HPV integration. I think I phrased that.
Yes, we have I'm in the cancer epidemiology branch, and so some of my colleagues have very big studies, like we have a study with Kaiser Permanente in California, where we get sample of all the women that they're screening for HPV or cervical cancer. And so we actually have a very large cohort of HPV sequences from women, exactly as you said, who are infected with HPV but don't have cancer, and those that have either precancer or cancer. One of the things we found is that we found that the E7 oncogene is essentially never altered in a tumor, whereas we found a number of rare amino acid altering variants in women who were infected that didn't get cancer. And that's led us to a study where we've actually studied those proteins in vitro and shown that virtually every one of them seems to be somewhat disabled. And so it looks like you can't change any amino acid in the E7 protein without somewhat disabling the virus. It can still be infectious and transmit from one individual to another, but it seems not to cause cancer. And that's important because it suggests that if we were to have a targeted therapy for HPV, that E7 would be the protein to go after, either by immune therapy, and then the virus can't change it to avoid the immune therapy, because if it changes it, it'll be a less effective virus. Or if we had some kind of small molecule drug that can inhibit E7, that can be very powerful. And then we also found some variants in the promoter region, which were also always found in women who had HPV but didn't have cancer, suggesting that, again, that those are somewhat attenuated forms of the virus. And those variants affect transcription factor binding sites, which we were to predict, but they're in a region of the regulatory part of the virus that had never really been studied before. And so that's pointed us to new transcription factors, which seem to be binding to HPV and having important roles either in gene expression or replication that we're going to study further.
Yeah, really interesting. There seems to be a pretty clear through line through a lot of this work about E7.
Yeah, and we're lucky to be here within a group that has, we have epidemiologists and groups running clinical trials with the vaccine and collecting samples. So there's sort of a wealth of expertise here that's great for me. I can tap into the DNA genomics part of all this, but have behind me the epidemiology behind it. So.
Well, we're about at time, but I'll sneak in one more question. Have you had a chance similarly to compare the cervical cancer data in the TCGA database that's you can get to through the CGC?
We are working with the TCGA data. Actually, the fellow I'm doing with that is, running his own command line program, so he downloads it separately. But we are interested in digging more into that. I was just looking at their database the other day, and you also have CCLE.
Mm-hmm.
I didn't actually see any of the cervical cancer cell lines that are part of CCLE on the database. So that would be an interesting question of, is that data there and I just couldn't find it? Or did that data not get uploaded yet somehow? Mm-hmm. But we are looking into that because we can dig further into the HPV sequences in TCGA and look at some of the same questions that we're answering here.
Yeah, cool. Well, thank you so much. I really appreciate you taking the time to tell us all about your research. Really fascinating story.
My pleasure, and thank you guys for the opportunity, and thanks for CGC to having this wonderful resource.
Yeah, thank you again, and have a wonderful afternoon, everyone.